Good morning, everyone. It's a pleasure to be here. I'm Davide Rovelli. I'm from the University of Parma, and this is my research project. I'm a PhD student, and this project is about the coffee leaf valorization through a metabolomic approach. So, before starting, I would like to thank University of Parma and Lavazza for funding this project, but also the International Coffee Convention for having me today here. So I would like to start with a simple question to introduce the topic. In particular, what do we know about coffee leaves today? So I give you some number to understand the impact of leaves in the coffee supply chain. So coffee in general produce more or less 2.3 millions of tons of solid residues every year. And for example, Brazil, that is the highest in production of coffee, is also the highest in the waste generation. And it's estimated that more than 80% of the waste produced in the country came from the coffee plantation. So in all these waste, leaves and twigs constitute approximately to 10 to 30% of the gross that can generate every year, and it's mainly produced by pruning of the plants. So why do we study coffee leaves? Why do we need to study them? I investigate a lot, and I find three main reasons to do that. The first one is the marketing, of course. We know that coffee leaves and its infusions are a traditional food in foraging countries, such as Brazil or Indonesia, etc. But also they have recently become a novel food in the European Union. This kind of product, so infuses or tea-like beverages, are also near to the products that are consumed in emerging markets. So this kind of product can be very similar to one that we know very well. And also, this kind of product allow us to have a diversification inside the coffee supply chain that is mainly based on coffee, of course. The second is the scientific point of view. So we know that this product is new, but this product can be used to produce nutraceutical. In particular, is a low-cost material, and it's rich in phytochemicals. But these phytochemicals are still underexplored in their potential, and so we need to understand better the fingerprint of these metrics. The last one is the social aspect. So we know that use byproducts can have a social aspect, important aspect inside the coffee supply chain, because we know that using them we can increase the income of the farmer, but also reduce the seasonal work inside the field, and also promote a circular economy and sustainability inside the coffee supply chain. So, metabolomic approach, what does it mean? So, if we start based on the fact that this kind of metrics is beneficial, we need to necessarily understand what is the profile of bioactive components inside these metrics. So we have developed this protocol. Is a single-step protocol that from a coffee leaf sample we are able to extract both polar and non-polar fraction of our leaves. And we use this fraction to make metabolomics, but also lipidomics, using an high-resolution mass spectrometry technique, but also low-resolution mass spectrometry technique. So this kind of approach provides us an holistic view of what is present or absent inside our metrics. During the last year, many questions arise, and I want to share with you some of them. So the first one is there any nutraceutical potential inside these leaves? How we can exploit or valorize them? How we can investigate the sensory properties of this kind of byproduct that can be used to produce infuses, so new product. If any there of impact in variety or origin in the chemical profile, but also there are compounds that are not yet been identified in our leaves. And finally, is there any form of chemodiversity? So to answer to this question, I would like to make an example that I'm sure that is familiar with all of you. So we can see coffee leaves as coffee beans. So we use coffee beans to produce, for example, an espresso. So imagine that we put these leaves inside a machine, a coffee machine that is, in this case, a Foodomics. So Foodomics, with targeted and non-targeted approach, provide us not coffee, of course, but data. But data are useful as coffee because coffee boosts our activity every day. They give us energy, and data give us much power to understand and to answer to those questions that I showed you before. So metabolomic data, in a nutshell, are these. So a long, long list of numbers, and our duty, in this case, my duty, is finding a sense, or in another word, find the answer to those questions. So I want to share with you some using of these metabolomics data with 3 applied example that came from my research activity. And I wanted to point out the potential of this technique to understand the potential of a byproduct, and also this kind of technique can be applied to other examples. The first one is the impact in the post-harvest production process of coffee leaves. So in this study, we investigate the quantitative determination of signature metabolites, but also we use the metabolomic approach to dive in the phytochemicals profile of coffee leaves. So we collect leaves and we treat them using a tea-like processing method. So we obtain an herbal leaf, a slight oxidation leaf, and high oxidation leaves. The aim was assess the suitability of metabolomics in the discrimination of commercial batches of these innovative botanicals. Our results are quite interesting because for the quantitative part, we assessed that the content of the main signature metabolites inside coffee leaves change according to the post-harvest process. But also the main chemical classes that are present inside these metrics also change. So we have a huge impact in the post-harvest process, and this is quite important if we consider that this process alter the composition of coffee leaves and also the bioactivity, but also the sensory potential of these metrics. Moreover, we need to standardization of producing coffee leaves, because also it is important if we consider that it is a novel food. So regulations set critical values of compounds such as caffeine that can be present in these kind of botanicals. And finally, we know that the mention of just coffee leaf extract can be misleading for the consumer. The second example that I want to share with you is a proof of concept of work that is still ongoing. So the discovery and the recovery of compounds. So we know that coffee leaves are discriminated from the beans for the presence of these chemical classes, so xanthones. And the mangiferin and isomangiferin are quite interesting because they was first discovered in another tropical crop, such as mango. So mangiferin and isomangiferin are quite interesting because they demonstrate many health perspectives. In particular, they exert protective effect against degenerative disease. And in particular, this compound can be very useful to investigate because they add an additional layer of benefits to the already rich phenolic profile that is present inside leaves. So the question is very simple. Are there other molecules related to them that we don't know about? So our previous study that I showed you before focused a little bit on this aspect, and we found that there are a lot of compounds, in particular this mangiferoxanthone A, that is a dimer of the monomer mangiferin, that exert an higher bioactivity compared to the other. And also their content is not significantly affected by the post-harvest production process. So we know that the xanthone family is much more complex than we think. So to investigate them, I want to show you this proof of concept of work, and is based and it starts from the collection of the leaves. So we collect leaves from Coffea arabica caturra, and in particular we clusterize the leaves according to their position in the branch. So we have mature, adult, and young leaves. We extracted them, and we use the mass spec data to build a molecular network. So a molecular network is based on the fact that molecules that belong to a chemical class share some functional group. And this group, when we obtain a mass spec data, is quite similar. So if we match these mass spec data to each other, we can clusterize them, but also we can keep out other molecules that are not correlated to this chemical class. So using these two platforms and some algorithms, I built this. This is a metabolomic network of mangiferin that I found inside my samples. And it's quite interesting because mangiferin is present, of course, but also there are a lot of compounds that was previously identified in coffee leaves. This is quite interesting because they can used in the future to understand what is the origin of this kind of compound, but also understand how to use better these leaves. I want to focus you also on their content. So, for example, mangiferin is much more present inside mature leaves, but their precursor, the homomangiferin, is much more present in the young leaves. So we have this changing inside our plants, in our branch in particular. So this is quite interesting, and it can be applied to other molecules that are present inside our sample. The last one is the map of chemodiversity. So plants produce a plethora of specialized metabolites, and they have a huge impact in our society. I'm sure that every one of you are familiar with this kind of molecules that came directly from the plants. So plants biosynthesize these compounds in response to environment, but also as a consequence of the growth and the development of them. So I collect leaves from Nicaragua, and in particular, in this study, I consider some varieties of Coffea arabica that belong to different genetic lineage. So Ethiopian landrace, Introgress, and F1 hybrid that have a huge importance inside the coffee supply chain. So the aim of the study was to explain the chemodiversity among different coffee varieties, but also understand what is the response in the phenotype and the genotype according to environmental stress in a climate change scenario. But also, I would like to understand how use coffee leaves, of course. Our preliminary data are quite interesting because they show very clear how this phenolic profile in the control sample is changed according to the genetic lineage, but also the stress sample, the stressed sample, have an evident change in the phenolic phytochemical composition according to this environmental stress. To conclude, I would like to share with you that discovering of bioactive components is a starting point to understand how use a byproduct, and is critical in the setting groundwork of this valorization and this upcycling. So if we combine a waste with an Omics platform like this, we can understand very well how to use them, and in particular we can find the way to discover and recover functional compound, but also use this technique to assess this byproduct for example, for their bioactivity, for their standardization, and their control in trade. Moreover, this useful information can add a layer of economic value to this byproduct, and we convert them from a byproduct to a new product. So I would like to thank all the people involved in the project, for my professor, Chiara Dall'Asta, also the Lavazza team, Adanucci and Bianca Serito, and finally, Christophe Montagnor, for his scientific contribution along the project. Thank you, everybody, and that's it.